We're set to go. Okay, good morning all. Can you hear me okay? Yeah, very good. Okay, so last week we looked at hematology, the components of blood, and hematopoiesis, how those components are formed in the bone marrow. Okay, so now hopefully we have kind of a basis upon which to continue today's two lectures. First hour, the lymphoid system looks at how the body defends itself, primarily the immune system, and it has a lot to do with lymphocytes. We didn't talk about lymphocytes hardly at all last week. And then in the second hour, we will be looking at where many of those components of blood lodge within specific organs and tissues within the body to carry out immune surveillance. Okay? Any questions before I proceed? So here's our learning objectives. Know the differences between physical, innate, and acquired defense mechanisms of the body. We'll cover all three of those. And of course, you need to know the cell types involved in each defense mechanism. Understand the mechanism of humoral versus cell-mediated immunity. That's basically antibodies versus cell-mediated immunity. Know the five major classes of immunoglobulins, i.e. antibodies, their function and where we find them. And understand the progression of antibody response and its relevance to immunization. I think that's particularly important in this day and age where there are anti-vaccine people who are completely ignorant of the benefits of immunization and are a danger to society because when they don't immunize themselves or their children, then they can spread diseases. And lastly, know the differences in the major histocompatibility complexes, their localization and interaction with specific lymphocytes. So that covers a lot, and so we shall first then review the three mechanisms by which the body can defend itself. Firstly, there are physical barriers that the body mounts to being invaded by foreign pathogens. Secondly, there are what we call innate or non-specific mechanisms of immunity, basically phagocytosis of foreign pathogens. And thirdly, there's acquired or specific immunity, which involves like making antibodies to specific pathogens or T lymphocytes, specifically recognizing foreign pathogens like viruses and mounting an immune response against them. So firstly, if we look at the physical barriers, we have things like tears, okay? So tears, when you get dust in your eye, you'll tear up sometimes and it will help to wash that dust, or it might be bacteria on the dust, whatever, out of your eye. Commensals are basically a word for bacteria that have a symbiotic relationship with the body to facilitate the functioning of the body. So we have commensals in the respiratory tract and the GI tract, as shown here. Of course, the skin, which we have already covered, is a physical barrier. We've done skin, haven't we? We haven't done skin? Oh, sorry, next week. I've done that at Georgetown. I get confused as to what I've taught here and there. Okay, the skin acts as a physical barrier to foreign pathogens readily gaining access to the body. And then we have low pH and commensals in the reproductive tract and the gut. Bacteria don't like an acidic environment, so by having a partial acidic environment in these particular locations, it slows down the growth of bacteria. In the nasal cavity, of course, we have cilia and also down the respiratory epithelium of the respiratory tract that can motion and help to expel pathogens like when you cough. So it's as shown here, acidic environment. And I talked about commensals, and also just by flushing of the urinary tract, like in the bladder. You know, if you're getting a bladder infection, I'm told if you drink a lot of fluid, that can help sometimes to flush the bacteria out. So these are physical barriers and exterior defenses that help the body in a limited way defend against invasion by foreign pathogens. The second mechanism by which the body can defend itself is through innate or non-specific immunity. So this involves primarily neutrophils and macrophages, whereby these cells can phagocytose, in this particular case, a bacteria. So when they come into contact with these pathogens, they will phagocytose them and obviously remove them from that location. Okay, so some of the cells that are involved in this non-specific or innate immunity include mast cells that will promote an inflammatory response. Obviously, neutrophils are phagocytic. Eosinophils can attack parasites. Of course, we know monocytes can differentiate into macrophages and a variety of other related cells like Kupffer cells, glial cells, osteoclasts, and so on. Macrophages are phagocytic, having a similar function to the neutrophils. And there's another type of lymphocyte called the natural killer lymphocyte, which can also destroy foreign pathogens in a innate or non-specific way. All right, so here's our natural killer cells. They represent, I think, around 15% of the lymphocytes in the blood. And there's kind of some interesting work that's been done on these natural killer lymphocytes. These are lymphocytes, right? Natural killer lymphocytes. It turns out that surgical stress reduces the number of natural killer cells in number and function in the body. So, I mean, this has been known. The fact that people have suppressed immune system after major trauma, so we consider surgery a form of trauma, obviously. Any sort of trauma, including surgery, has been shown to drastically reduce the number of natural killer cells. And so we see this post-operative drop in natural killer cells, usually peaking after around 3 days, and it takes about a month to recover the number of natural killer cells. So it's good to be aware of that. And also because there's this reduction in natural killer cells, that may in fact facilitate the seeding of metastatic tumor cells. So if a person is having surgery to remove a tumor, remember when the surgery is a form of trauma, their natural killer cell numbers will drop post-operatively, and during this 3 months, this month or so, when they have a reduced number of natural killer cells, if there are some single tumor cells that escaped the surgery, they may be able to escape immune surveillance because the natural killer cells are no longer there to attack them. So that's kind of that first month after surgery is a kind of a region of risk, if you like, a period of risk where the immune system is suppressed, particularly in regard to natural killer cells. So that's good to know. So since surgical stress reduces the natural killer cell numbers, obviously you want to limit surgical stress, and thereupon comes recent advances in surgery where a lot of the time now they've been able to implement robotic surgery. Okay, robotic surgery. So I had surgery for prostate cancer this time last year, where they took out my prostate gland, and it was done with robots. Okay, and there's I had 6 little half-inch slits in my abdomen, and they put instruments in through those slits, and everything's done by the surgeon behind a screen. So I was on the table, unconscious. There's some nurse assistants in there to help move some instruments around. And the surgeon is behind a glass screen, sitting on a nice comfortable chair. He doesn't have to scrub up anymore, right? And he can sit down. So he's just moving these instruments around behind this glass partition, doing the robotic surgery. And of course they've got cameras in there and all sorts of stuff, and surgical instruments and lights, and it's all done. But the thing is, the amount of surgical stress was very much limited because they're just little slits. In the old days, they would do abdominal surgery for prostate cancer, where they do a big slit and then they open you up, and take this like major, and that would take like months to recover. Well, I was walking around the next day, and after a week after they took out the cannula, I was up and around and on my way. So, I mean, that was one way to, through robotic surgery, limit surgical stress and increase the recovery time for the patient. The other thing that I'm told is because with this robotic surgery, in addition to it being a major benefit to the patient by reducing surgical stress, it's a major benefit to the surgeon because the surgeon's sitting on his butt in a nice comfy chair. He doesn't have to scrub up. And so, because a lot of the times, in the old days when surgeons are standing up, that's physically demanding. If they're doing surgery for hours and hours, it can be very demanding on the surgeon himself. But obviously, if you're no longer standing up and walking around, if you're just sitting down in a nice comfy chair in your sweats instead of your scrubs, they've told me it's extended the life of many surgeons' careers by about 10 years. So again, it not only helps the patient, but it also helps the surgeon extend his career. So there you go, natural killer cells, reduced after surgical stress, and you want to reduce surgical stress, hopefully these days with a lot of robotic surgery. Okay, so that was the innate or non-specific immunity. Now, the third type of immunity, acquired or specific immunity, we're going to spend the rest of this hour upon. So acquired or specific immunity typically involves an antigen-presenting cell, such as a macrophage or some of the other components that have differentiated from a monocyte into macrophages, Kupffer cells, glial cells, osteoclasts, and so on. What happens is, say, a bacteria comes into contact with a macrophage and it gets degraded in lysosomes, chopped up, and then a little fragment of that bacteria then is bound to a protein complex called the major histocompatibility complex, and it is presented to a lymphocyte. The lymphocyte has on its surface IgDs, which act as receptors, and if the variable portion of those IgDs is complementary to this fragment of the bacteria, that will activate that lymphocyte. If it is a T lymphocyte, it will produce more T cells for cell-specific immunity. If it's a B lymphocyte, it will mediate humoral immunity, where the B lymphocyte differentiates into a plasma cell and produces lots of antibodies that are specific to that bacteria and then can remove the bacteria. So there's two components to acquired immunity. There's cell-mediated immunity involving T cells, and humoral immunity involving B cells differentiating into plasma cells becoming activated to produce antibodies. So this is a relatively recent evolutionary adaptation. If we look at when acquired immunity occurred, maybe about 400 million years ago, because we know that sharks were the first type of vertebrate to have this rearrangement of immunoglobulins, which provides for specificity against foreign pathogens. Either in B cell immunoglobulins or the T cell receptors have a rearrangement of genes, the rearrangement of a component of the receptor against foreign pathogens. So it's relatively recent. All right, so let's look at some of the functions of the lymphoid system and how they protect against the body and provide immunity. Well, firstly, if we're going to have specific or acquired immunity, we have to be able to distinguish a self-antigen from a non-self antigen, okay? So non-self, that would be bacteria, and self would be, all the normal cells of the body. So when the immune system is activated, we have to recognize foreign material, foreign microorganisms. Sometimes the immune system will recognize cancer cells as foreign, and that actually is a relatively new field of research and development for treatment, where now they're able to activate, in some cases, the immune system to fight and destroy cancer cells. And it's also very important in terms of organ transplants, because obviously if a person gets an organ transplant from anybody other than an identical twin, the body will recognize that transplant as foreign. So if it's a foreign pathogen, foreign transplant is going to try and reject that transplant, right, as if it was like a bacteria or anything else. So in those cases, people that have organ transplants have to take drugs to suppress their immune system so that the body won't reject the organ transplant. Okay, so types of acquired immunity. Humoral immunity involves B cells differentiating into plasma cells secreting antibodies, and cell-mediated immunity involves cytotoxic T cell killing of cells that have been infected with, say, viruses. So here's a comparison of humoral and cellular immunity. So what we see here is that the foreign pathogen, let's say it's a bacteria, comes into contact with a B cell, which has on its surface IgD molecules, immunoglobulins, which have a variable portion, which may be complementary, may recognize this foreign pathogen as foreign, in which case that will activate that B cell to differentiate into plasma cells and produce antibodies against that foreign pathogen. In addition to B cells differentiating into plasma cells, a few of the cells will become memory cells, which can reside for, depending upon the type of immunity, months, years, or decades in the body, where if you ever get re-exposed to that pathogen, the body can mount a secondary, much more rapid response against that pathogen. So basically, humoral immunity involves the production of antibodies. That's the bottom line there. The other form of acquired immunity is cell-mediated immunity. In this particular case, it involves cytotoxic T lymphocytes, which have on their surface a receptor complex which can come into contact with a cell, like in this case, just a normal cell that has been infected with these viruses, these little green dots of viruses. And if these viruses are bound by what we call the major histocompatibility complex 1 on all cells, this MHC1 presents the virus to a T cell, and if the T cell receptor, which also has a variable portion, recognizes the, in this case, virus as a foreign pathogen, that will cause that cytotoxic T cell then to destroy that infected cell, in this case a virus-infected cell. So that's cell-mediated immunity, where the cytotoxic T cell directly destroys an infected cell, or humoral immunity, where B lymphocytes differentiate into plasma cells, produce antibodies against the bacteria. So where are these lymphocytes coming from? So we talk about the immune system as having central lymphoid tissues and peripheral lymphoid tissues. The central lymphoid tissues basically are twofold. The B cells are derived from the bone marrow, and the T cells start out as stem cells, migrate to the thymus, where they undergo maturation as thymocytes and then become T cells. And these, both the T cells and the B cells, have, that is the origin, those pluripotential stem cells, remember, from hematopoiesis, that reside in the bone marrow. If those stem cells are going to differentiate into B lymphocytes, that will occur in the bone marrow. Now, the research involving B lymphocytes was actually carried out originally in chickens, okay? And in chickens, the B cells that differentiate into plasma cells, which is antibodies, reside in what is called the bursa, which is a gland under the cloaca of the chicken. So, do you know what a cloaca is? Okay, so birds, reptiles, amphibians, fish, have a cloaca. There's one common urogenital opening. There's no separation between the excretory, like large intestine in us, and the reproductive exits, like the vagina or the urethra, of the men's penis, right? So in mammals, those two are separated. There's the genital opening and the excretory opening. They're separate in mammals, but in birds, reptiles, fish, they converge inside, and there's one common exit called the cloaca, and that cloaca in Latin means sewer. Okay, so they combine into one sewer exit, the cloaca. So underneath the cloaca, in chickens, you'll find a bursal gland, and this is where they were able to do all the research on B cells differentiating into plasma cells producing antibodies. Fortuitously, bursa starts with a B, and in humans this occurs in the bone marrow, so then they became B lymphocytes, right? B for bursa in chickens, B for bone marrow in humans. So the B cells then are released from the bone marrow in humans, where they can circulate throughout the body and in some cases differentiate into plasma cells to produce antibodies. In contrast, the thymus produces the cytotoxic T cells. In this case, the stem cells migrate to the thymus where they undergo further maturation into T cells, and then they can be released into the circulatory system. So then we consider the bone marrow and the thymus to be the central lymphoid organs, and everything else, all the other tissues and organs, are peripheral lymphoid tissues. So now let's go back and look at, in great detail, humoral immunity. So humoral immunity means production of antibodies. So humoral immunity has as its basis B lymphocytes that originate in the bone marrow and are released into the circulatory system, where if they become activated, become plasma cells and produce immunoglobulins to mediate what we call the humoral response, basically an antibody response. Okay, so here's our bone marrow, and it releases the B lymphocytes into the circulatory system. Some of those B lymphocytes will become plasma cells to produce antibodies, and a few of them will also become memory cells, so that if you ever get infected again by that same organism, the body can mount a much more rapid response. Now it turns out, pardon me, B cells can also sometimes be produced in the liver. Okay, so we know for sure that it occurs in the fetal liver. Remember the hematopoiesis lecture where we have the hepatosplenothymic phase of hematopoiesis, and during the fetal phase, hematosplenothymic phase, hepato, liver, spleen, spleno, spleen, thymic, thymus, in the second trimester predominantly. So in the second trimester, the fetus is producing lymphocytes predominantly in the liver, and you can actually have that reactivated at a later time in adults if the bone marrow is damaged through disease, or you might have got zapped in Fukushima, next to when you get exposed to a lot of radiation, destroyed the bone marrow. In those particular cases, the liver has a memory of its fetal life back in the second trimester, has this genetic memory where it can reactivate lymphopoiesis in the liver. It occurs in certain diseases. But in most cases, of course, it occurs in the bone marrow where you get these cells produced. Okay. So once these B cells become activated, they become plasma cells, okay? And you remember a plasma cell, we know from the connective tissue lecture, the different cellular components of connective tissue. We recognize plasma cells because they have an eccentric nucleus, i.e. off-center. Sometimes we can see heterochromatic arrangement of chromatin looking like a cartwheel arrangement, but not in this one. And also we have an abundance of rough endoplasmic reticulum, which of course we have to have a lot of rough endoplasmic reticulum to make the immunoglobulins. So here this is just reminding us that the rough endoplasmic reticulum in plasma cells is producing the immunoglobulins as shown here and then secreted from the plasma cell. So if we look at the structure of immunoglobulins, i.e. antibodies, immunoglobulins, the basic structure of all immunoglobulins is shown thus. We have two long chains, we have two short chains or light chains, heavy and light chains. The two heavy chains are bound together by disulfide bonds, and one heavy and one light is bound together by a disulfide bond. Most of the heavy and the light in pink is a constant portion, and significantly at the end of the light chains and the heavy chains we have what is called the variable portion. This sequence of amino acids in the variable portion varies from B cell to B cell, and if that variable portion turns out to be complementary to some sort of foreign pathogen, that will induce that B lymphocyte to undergo clonal expansion to produce antibodies, so that then that variable portion can bind to that pathogen and remove the pathogen and mount a humoral response. So how do we get this variability? How do we get this variability in the variable portion? Well, it turns out this occurs by what we call somatic recombination. So basically, the light chain has these are exons of DNA, right? So you have these different sequences of the variable portion and different sequences of the joining portion, and they get shuffled. So you might get, V1, V2, V3, J2, or it could be V2, J4, and so on. It's basically a random event where you're getting shuffling of these different exons to give you these different sequences. And purely at random, if one of those sequences turns out to be complementary or sufficiently complementary to a foreign pathogen, that is the go signal for that particular lymphocyte then to undergo clonal expansion to produce antibodies. Similarly, the heavy chain has a variable joining, and there's also another diversity joining exon, right? So you've got the DNA exons transcribed into the RNA, which is then translated into the protein with these different sequences. And this is purely a random event. The body is generating constantly these, undergoing this somatic recombination of these variable diversity and joining regions in the B and the T cells. B cells for the antibody, this occurs in the T cell receptor. And if those final products, the variable portion is complementary to a pathogen, that's the go signal to then undergo further clonal expansion to attack that pathogen. That's a random event, but it's happening constantly. So here are our different 5 classes of immunoglobulins that have this common structure that we just looked at. The most frequent immunoglobulin is IgG. It occurs as a monomer. Most of the antibody in the body is IgG. We see a lot in the blood and lymphatic fluid. And it can neutralize antigens just by binding to them. We also see immunoglobulin M, which is a pentamer. There's 5 of these basic monomeric subunits bound together with these together shown here. And this is actually the first antibody that's produced in response to an immune response. For instance, when you get immunized, usually it's IgM that comes first in greater abundance than IgG, and we'll look at that momentarily. Then we have IgA, which is considered the secretory immunoglobulin. It acts as a dimer bound by a protein. And the IgA, we call it a secretory antibody because it is secreted onto the surface of mucosas. So, for instance, along the GI tract or the respiratory tract, it will be secreted across the mucosa into the GI tract lining the surface, or the respiratory tract, where it can readily come into contact with pathogens early on before those pathogens can penetrate across that mucosa into the blood or the connective tissue. IgD is very rare, and it acts as that receptor on the surface of the B cells that I was talking about. And IgE, even rarer, has a similar function as a receptor on the surface of basophils and mast cells. Okay? So they're the 5 major classes of immunoglobulins. So let's look at what happens when you get immunized. Okay, who has had their flu shot this year? Very good. Okay, very good. The rest of you, tomorrow, go get it. Because if one of you gets sick, you're going to spread it to everyone else. This is what happened when you get a flu shot. This is the flu vaccine, which is inactivated virus. Is usually these days it's quadrivalent, meaning 4 different strains, right? 4 different strains of virus that's been inactivated. And so you get injected with the vaccine, and it takes a little while, up to 10 days usually, during the first immune response for the body to react. And then during this primary response, what happens is you get more IgM produced than IgG, okay? And there's a certain level of the response. Now, then that declines somewhat. But during this primary response, not only did the body make immunoglobulins, it also made memory cells, which is the critical event here, because the memory cells have memory of being exposed to this vaccine, this antigen. So next time around, the 90% of you that haven't been vaccinated sneeze on those 10% that were vaccinated, and those 10% that were vaccinated get exposed to this virus. But because they have these memory cells, there's a much shorter lag time for the body to respond, okay, on the second exposure, much shorter lag time. During the secondary response, there's a much greater amount of antibody produced because these memory cells can kick in much quicker. They've already been exposed to the vaccine, so they're available to rapidly respond. It's a rapid response team, if you like. They produce a much greater amount of antibody, and during the secondary response now we get much greater amounts of IgG compared to IgM. So this is the basis of immunization. Basically, the first exposure gives you this primary response significantly with memory cells. The second exposure to the real thing, okay, gives you this much greater response because of the presence of memory cells. So that's why and how immunization works. So I recommend you all get immunized. Okay, so how do antibodies work? So this is the humoral response. This is listing 5 different mechanisms by which antibodies can eliminate a foreign pathogen. So the first one is agglutination, in which the antibodies bind to our bacteriaceae. So this reduces the amount of free pathogen floating around in the body. The second is opsonization, where the binding of the antibody to the bacteria then tags it for phagocytosis by our macrophage. The third is neutralization, where again the antibody binding to our pathogen then essentially inactivates the toxin because it's surrounded by antibodies, so the toxin or the pathogen doesn't have ready access to cells. The fourth is cytotoxicity. In this case, the antibody is acting as a bridge between, in this case, our eosinophil and our helminthic pathogen, the worm, pulls them close together, if you like, holds them close together so that then, remember, the neutrophils can secrete those neurotoxins, can kill our foreign helminthic worm pathogen. And the fifth is the complement cascade. So in the blood there's a series of proteins called the complement cascade, and in this case the binding of the antibodies to the initial component of the complement cascade initiates the cascade, whereupon the complement cascade will produce a complex including a transmembrane channel attack protein, which then can rupture, in this case, the bacteria and destroy the bacteria. So there's 5 different ways in which antibodies can promote the humoral response to destroy foreign pathogens that may be invading the body. Okay, so that was B cells and humoral immunity producing antibodies. Now let's talk about T cells. T cells, i.e. cytotoxic T cells. Remember, they mature in the thymus. They're released from the bone marrow as pluripotential stem cells, make their way to the thymus, where these T cells undergo maturation. And the T cells have on their surface a receptor. That receptor complex has that variable portion of protein that will be looking for a foreign pathogen, complementarity. The T cells, we have our cytotoxic T cells carrying out destruction of pathogens. We also have the memory cells, just like we had for the B cells. And there are some other T cells that are helper T cells that can promote the activity of other components of the immune system. And this activity of the helper T cells a lot of the times is facilitated by cytokines, sometimes also called lymphokines. Okay, and this is a big family of growth factors that have variable effects on the immune system. That is something that you may or may not learn in immunology lectures. We're not going to cover it here because it's a whole lecture unto itself. Now it turns out T cells, obviously once you activate an immune response, once it finishes its job, it has to be down-regulated, right? You don't want it running wild. So the way that T cell immunity is down-regulated is through what is called the PD-1 cell surface receptor. It's expressed on T cells. So PD-1 and its ligand that binds the receptor down-regulate T cell mediated immunity. Okay, so it's a brake on T cell activity. So drug companies recently have found drugs that will inhibit the PD-1 receptor and its ligand. Okay, so what they're doing is they're inhibiting the brake. So if you inhibit the brake, sometimes that works the same as turning on the accelerator, right? Inhibit the inhibitor allows reactivation. So this actually has been I think it's called Keytruda. I've seen it advertised on TV for lung cancer, Keytruda, something like that. So this has actually been extremely successful for cancers that express the PD-1 receptor. And this is what President Jimmy Carter is getting, or was treated for. He had skin cancer, he had a melanoma that had spread to the brain, and usually once you get any sort of brain cancer, the prognosis is maybe a year or so. He was treated with this and it completely got rid of it. It was like a miracle almost, like you never see that for brain cancer. And now they're using these PD-1 inhibitors for any and all types of cancers. They look at the cancer cell, the pathology, and they're going to see whether they express the PD-1 receptor. If they do, they can treat them with this drug. I think it's called Keytruda, something like that. And it's having fantastic results, extending the life indefinitely for some people. Like, do you guys know who President Jimmy Carter is? You heard of him? He's pretty old now. He's in his 90s. It's lasted a long time. Complete remission, or some people will extend their lifetimes. Was there a question? Yeah. Mm-hmm. No, the B cell doesn't have the receptor, not the same receptor as the T cell. The B cell has the IgD on its surface. Because it's an immunoglobulin, IgD, it has that variable portion. If you go back to that slide where I showed you the 5 different classes of immunoglobulins, it'll show you their IgD acts as a receptor on B cells. And in IgE acts as a receptor on mast cells and basophils. Okay, so that's a little bit of information on T cells. So now let me give you a little bit of background on milestones in therapy. These are the different types of therapies, different categories of therapies that can be used to treat people, kind of in order of their development throughout the century. So surgery, obviously, has been around since antiquity, right? Since the days of the Roman Empire and the Greek Empire, when people would have diseases, they could be treated surgically. Of course, the big breakthrough with surgery was the introduction of anesthetics, because a lot of people would die from the surgery. With the introduction of anesthetics, you can carry out anesthesia and the patient hopefully doesn't know about it. After that, more recently, right, we have radiation, particularly for treating cancers. So this was a result from Marie Curie. You guys know who Marie Curie is, right? She discovered basically radiation, radioactive decay, and then she found that her and later on, I guess, development, people found that it could be used to destroy cancers. So then after that we have endocrine therapy. I think one of the first people to use this was Charles Huggins. He was the guy that discovered that through removal of testosterone by castration, basically you could cause prostate cancer to undergo remission. I thought this was fantastic, but then it turns out after about 2 years the cancer cells become androgen independent and come back again. But at least that was the first idea of endocrine therapy. And of course now you have endocrine therapy like for breast cancer, where they have agents that antagonize estrogen receptor, progesterone receptor, and HER2 and things like that. So endocrine therapy. Then kind of related to endocrine therapy is chemo. Like a lot of chemotherapy, the original chemos were drugs that would cause microtubules to dissolve. So we know during cell division, right, we have the mitotic spindle, right, mitotic spindle during metaphase. So the idea is if you can destroy cancer cells divide more quickly than other cells of the body. So the idea is if you can inhibit mitosis, you can inhibit the more quickly dividing tumor cells more so than normally dividing cells. So drugs that dissolve the microtubules would destroy the mitotic spindle, so then those cancer cells can't divide, compared to normal cells. Of course, cancer cells being what they are, they get around it, but that was the first chemotherapy, and then of course there's many different kinds. Gene therapy has come along recently, just starting to be used, very complicated, not so easy to apply, where people are trying to put back normal genes back into diseased patients to replace some sort of congenital disease. Okay, it's being used experimentally, and now there's just a couple of treatments where it's actually being used for certain diseases. Along with that comes stem cell therapy, where the idea is you can induce a stem cell to differentiate into the normal cell and replacing some sort of defective cell. And of course now immunotherapy, like that PD-1 that I just talked about, right? The T cell PD-1 receptor. So now immunotherapy is being really explored in great detail. This is like the big thing right now in clinical research to try and develop new ways to treat cancers, in particular with immunotherapy. It's kind of, immunology is pretty complicated, so it's taken them a long time to figure out how a lot of this stuff works, but now they're getting a grasp on some of it, and now the pharma companies are really going gung-ho trying to develop and discover new immunotherapies to treat patients. So that gives you like a history in chronological order of the different types of therapies that have been applied to treat patients, a lot of it related to cancers over the years. Okay, so T cells. We did B cells. T cells, remember, originate as a stem cell in the bone marrow, migrate to the thymus, which then is considered the central lymphoid tissue, where there's T cells undergo maturation and become T lymphocytes released into the blood. We have cytotoxic T cells that directly lyse cells. We have helper T cells that can stimulate the immune response. Suppressor T cells that are going to have PD-1 on them. And of course we always want our memory T cells for secondary activation on a subsequent exposure. So cytotoxic T cells, as I mentioned, have a T cell receptor complex. This is just a schematic for a T cell receptor complex. And this T cell receptor will have, part of it, will have a variable portion, okay, of amino acids. And then we have our antigen presenting cells. It can, in this case, it's an MHC. It has on its surface a major histocompatibility 2 complex, which is a complex of proteins that will bind an antigenic fragment, shown here. So the antigenic fragment, a chunk of bacteria or a virus, whatever, held by the MHC is presented to the T cell. And if the T cell receptor recognizes this antigenic fragment as foreign, that will cause that T cell to become activated. Okay, so MHC complexes come in 2 major flavors. All cells contain MHC1. So the MHC complexes, these are the protein complexes that are binding the antigen, the foreign pathogen, piece of a pathogen, whether it's a virus or a bacteria or whatever. So all cells have on their surface MHC1, and these cells, all cells containing MHC1, can interact with cytotoxic T cells. Sometimes they're called CD8 positive cells, cluster of differentiation 8 positive cells. It's the same thing, CD8 positive or cytotoxic T cell, same thing, synonymous. So all cells have MHC1, and they can interact with cytotoxic T cells. In contrast, MHC2 is restricted to antigen-presenting cells, such as macrophages, Kupffer cells, glial cells, and so on. All those different cell types that can differentiate from a monocyte. And so these antigen-presenting cells usually have the bacteria pass through lysosomes, where it's chopped up, and a fragment of that bacteria then is bound to the MHC2 complex and presented to helper T cells in this case. The helper T cell, which we call CD4 positive and helper are the same thing, synonymous, then can actually release a number of cytokines to promote differentiation of B cells into plasma cells to produce antibodies against that pathogen. Okay, so this shows you some examples of how this works. So here's our helper T cell, CD4 positive. Since it's a helper T cell, it will interact with a antigen-presenting cell, such as a macrophage, containing upon its surface the MHC2 complex. The bacterial fragment is presented to the helper T cell. The helper T cell recognizes the antigenic fragment as foreign. It will produce interleukins or lymphokines, cytokines. All three things can be synonymous to activate B cells to produce antibodies, become plasma cells, produce antibodies, and we always want our memory B lymphocyte. And the other mechanism is cytotoxic T cells, CD8 positive. So here is our target cell that's been infected with, let's say, a virus. A bit of that virus is bound to the MHC1 complex and presented to the T cell. And if the receptor component of the T cell is complementary to that bit of virus, then that cytotoxic T cell will become active and it will destroy that target cell. And it destroys that target cell through a couple of mechanisms. It can produce perforins, as the name implies. These proteins can create perforations in the plasma membrane of the target cell, so the target cell will lyse. Or they can produce a Fas ligand, which will bind to Fas, which will promote apoptosis. So Fas and the Fas ligand are a member of the death receptor family. There are proteins on the surface of cells. There's at least 6 or 8, can't remember, different types, that are called death receptors that when activated cause a cell to undergo apoptosis. So apoptosis is cell-mediated cell death. They involute. Okay, so the two types of cell death is necrosis and apoptosis. So necrosis is kind of like murder. You know, if you cut your finger with a knife, it ruptures the cell, it's necrotic cell death. Apoptosis is where the cell doesn't actually rupture per se, it causes the cell to destroy itself from within. The DNA becomes fragmented, it kind of involutes and it starts to phagocytose itself from within. So it's kind of like a suicide from within, so that's apoptosis. So then the cytotoxic T cells can activate this Fas-mediated apoptosis to cause the target cell to destroy itself. Okay? So that's how cytotoxic T cells work. And this just shows you the complexity of the MHC complexes. This is MHC2 and MHC1. You can see how many different gene products are involved in the creation of this complex. Okay? Okay, so some people have congenital defects in their immune system where the B cells and T cells are not active. So here's an example. This kid, David, more than a decade ago now, had severe combined immunodeficiency, combined because there's B cell and T cell involved. So he lived in this bubble, this plastic bubble, okay? He couldn't be exposed to the air, because if we just breathed the air and got infected with any sort of bacteria, as we breathe, we're constantly breathing in bacteria. Any sort of the most slightest infection would kill him, because he'd get infected with a bacteria or a virus and he couldn't mount an immune response, so he would die. So he had to live in this plastic bubble, which was sterile for his life. I'm not sure what happened to him. Okay, so it turns out that the CD4 positive helper T cells are the cell that is destroyed by the AIDS virus. Okay, so if you look at the etiology of an AIDS infection in a person that is not being treated, what we see is initially upon exposure in the white dot to the AIDS virus will infect the helper T cells and start to kill them. So over time, the number of helper T cells gradually declines. And as this occurs, the number of viruses crashes, but then you get to a critical point where the number of helper T cells is so low that they're no longer facilitating an immune response against the AIDS virus or anything else, so that then the virus can start to take over, and at the same time we get what are called opportunistic diseases. So when in the late 70s, early 80s, when the HIV first appeared in great numbers in the US, physicians didn't know what was going on, because these patients would show up at the clinic with diseases that they were extremely rare, that they never saw, like Kaposi's sarcoma, which is a skin disease. You know, these are things that they would see maybe occasionally in like Africa or some tropical country or something or other, but we're never seeing them in the clinic in the US. But all of a sudden all these patients were showing up with these crazy diseases because these were opportunistic diseases, okay, and they didn't know what was going on, and that would eventually kill them. So it wasn't the virus per se that was killing them, it was the fact that their immune system was suppressed and other diseases were opportunistically then arising and killing the patient. So that's how the AIDS virus typically will run its course if a person isn't treated. These days, of course, they have drugs that suppress the AIDS virus, so their lifespan is extended indefinitely. Okay, that's the first hour. Here's our question for this hour. Which of the following is correct regarding T lymphocytes? T lymphocytes, okay? Which of the following is correct? I'll give you a minute to think about it. Okay? Have you guys guessed an answer? Was that your answer? C? Do you want to ask me why it's not one of the others? Okay. It's C. Okay. There you go. All right. So let's take a 5-minute break, and then we shall move on to the second lymphoid lecture. Okay, so second hour on the different types of tissues and organs that the body houses, containing these different components of the immune system, primarily the lymphocytes, but also a couple of the other formed elements of the blood. So things to learn: know the different components of the immune system, the different organs, tissues, things like that. Be able to recognize them, okay, and be able to tell where different types of tissues and organs reside within the body and in different areas of different lymphoid tissues and organs. Okay, be able to identify them, okay, be able to distinguish one type of lymphoid organ or tissue from the other. That can be confusing sometimes because it's a whole bunch of lymphocytes. Sometimes it's not easy to tell. And obviously know the function of the different areas where these different lymphoid tissues and organs occur. So this is an overview of the different lymphoid tissues and organs of the body and classification based upon central and peripheral. So you will recall the two central lymphoid organs are the thymus and bone marrow. Thymus produces T cells, T for T cells, thymus, B cells, bone marrow. Everything else is peripheral. So we can have single cells, sometimes called the mucosa-associated lymphatic tissue. So mucosa is, lining of the GI tract, respiratory tract, reproductive tract, right? Mucosa. And you can have single cells. Sometimes you can have an aggregation which is somewhat diffuse. and it can be transitory. It comes and goes. Sometimes it's not so transitory, it's aggregated, like tonsils. And the function of all of these is antigen surveillance. So in the unencapsulated tissue, we have antigen surveillance of the mucosal surfaces, the lining of the GI tract, respiratory tract, reproductive tract. And in the encapsulated organs like lymph nodes and spleen, lymph nodes filter the lymph, the spleen filters the blood. So in all cases, whether it's filtering the lymphatic fluid, blood, or you have cells in the mucosal surfaces, the function is antigen surveillance. In other words, the immune system is on the lookout for any foreign antigen, surveilling for any foreign antigen in which it may mount an immune response. So B cells and T cells are differentially localized to differing degrees in different tissues and organs. So in lymph nodes, B cells aggregate in the cortex and the medulla, whereas the T cells are in the paracortex of the lymph node. This will be more meaningful to you once we do these specific organs and tissues. In the spleen, B cells are in the lymph nodules, whereas the T cells are aggregated in the periarterial lymphatic sheaths of the spleen. In blood and lymphatic fluid, most of the lymphocytes are T cells, not so many B cells in the fluid. Okay, mostly T cells, not so many B cells. Conversely, in the mucosal tissues, for instance in the lamina propria, underlying the mucosal tissues, most of the lymphocytes are B cells, not so many T cells. So you can see then that B cells and T cells are not evenly distributed. They aggregate to differing degrees in different tissues and different locations within organs. So here is a slide showing you the different types of lymphoid tissues with increasing complexity. So we can start out with our single circulating lymphocytes. If it's in the blood, it's going to mostly be T cells. Sometimes we can have an aggregation of those lymphocytes in tissue, so it'll be diffuse, and sometimes they can form what we think is a nodule. Then we get aggregated nodules, which are not completely encapsulated. In other words, there's not a complete connective tissue capsule around them, so we call them aggregated nodules as opposed to nodes. Nodules are incompletely encapsulated. We see aggregated nodules in tonsils, Peyer's patches, and appendix. And then we see specific organs which are fully encapsulated. When encapsulated, I mean a connective tissue capsule surrounding the whole organ. We see that in bone marrow, thymus, lymph nodes, and spleen. So this is a slide showing increasing complexity, and we see this progression of single cells to increased complexity of different organs during evolution of the vertebrates. You see in the most primitive vertebrates, you see GALT. GALT is the same as MALT. This is GI tract associated lymphatic tissue, same as MALT, mucosa associated lymphatic tissue. So in these organisms, you don't have organs, you just have loose connective tissue containing some lymphocytes. Then you get increased complexity with some different organs like that as you increase the complexity of the vertebrates. And then finally, in terrestrial vertebrates, we finally get encapsulated organs, and lymphoid organs was bone. Okay, so there's a progression of complexity, just as we saw a progression of complexity in the previous slide. So now let's go back and look at these in turn. So circulating lymphocytes. So circulating lymphocytes in the blood are mostly going to be T cells, and we'll get a smaller percentage of B cells and a smaller percentage of the natural killer cells. But most lymphocytes in the blood will be T cells. Then we can get diffuse lymphoid tissue. So for instance, in the GI tract, see all these little dots here? These little dots are nuclei, mostly of lymphocytes. Now, because we are now in the mucosal tissues, most of the lymphocytes in the mucosal tissues will be B lymphocytes, right? T in the blood, B in the tissue. Okay, in the lamina propria. Remember, lamina propria is a general term for the jumble of connective tissue that underlies most mucosal surfaces, such as the lining of the GI tract, respiratory tract, reproductive tract. And it turns out that the B lymphocytes residing in the lamina propria secrete mostly IgA. Okay, IgA. IgA we call secretory immunoglobulin. So what happens is that these plasma cells differentiating into, or B lymphocytes differentiating into plasma cells secrete IgA. And this IgA is taken up by, in this case, the lining of the GI tract, and they are transcytosed across the epithelium into the lumen of the GI tract. Now, normally, if a protein is taken up into the cytoplasm of a cell, it may, pardon me, be targeted for lysosomal degradation. So what happens with IgA is that a secretory component is added by the epithelial cells itself, these little spikes, added to the IgA dimer to protect it from degradation by lysosomes. So then that IgA dimer containing the secretory component makes its way across the cytoplasm of the GI tract intact, and once it reaches the apical surface, the secretory component is clipped and the IgA dimer is released into the lumen of the GI tract, whereupon it's readily available to interact with any foreign pathogens that you may have ingested during the normal course of events. Okay, so IgA, called secretory immunoglobulin, secreted across mucosal surfaces such as the GI tract, respiratory tract, reproductive tract. So that's diffuse lymphoid tissue. Now, solitary nodules consists of an aggregation of diffuse. So see all of these dots here, these are nuclei of lymphocytes, so this would be considered all diffuse. As the name implies, it's diffuse. But sometimes these diffuse lymphocytes may aggregate, as shown here, to form a solitary nodule. It kind of looks like a nodule. Now, nodule and follicle are synonymous, and it's nodule and not a node. A nodule meaning there's no connective tissue circumscribing the no connective tissue layer around the nodule. Now, sometimes we can make out a darker ring around the outside and a lighter center. So the ring we call a mantle, the lighter center, the germinal center. In that case, if we see a mantle and a germinal center, that's called a secondary nodule. Secondary nodule appears thus because the germinal center contains more plasma cells, and you'll recall plasma cells contain an abundance of cytoplasm making immunoglobulins. So since you've got more cytoplasm, this germinal center stains lighter. So we would consider this a secondary nodule because we can see a mantle and a germinal center. If we didn't see a germinal center, if this whole thing was uniform in density, just like the mantle, then that's a primary nodule. That nodule hasn't been activated yet. Okay, so sometimes we see aggregated nodules in the tonsils. Well, we do always see them in the tonsils. Here are the locations of the various tonsils in the oral cavity. You don't have to remember any of this. You don't have to remember the names of the tonsils, okay? Just showing you that they surround the oral cavity so that as you breathe and ingest, they are readily present to interact with any pathogens that you may be breathing in or ingesting. And here's an active nodule, so an aggregated nodule of the tonsils. So this is all mostly lymphocytes. There's a partial capsule on the basal surface, as shown here, but on the apical surface, there is stratified squamous non-keratinized epithelium, i.e. of the skin. So that's not connective tissue, that's skin. So then it's not fully encapsulated. Okay, it's partially encapsulated with connective tissue at the base, but on the surface there's no connective tissue, it is skin. So then we say that the tonsils are aggregated nodules because it's not fully encapsulated. And you can see these aggregations of nodules. You can see diffuse lymphatic tissue and these secondary nodules. You can see a germinal center and a mantle around each of these. So these are activated nodules that have responded to some sort of infection and are producing, because they're producing IgA, right? The secretory IgA is going to be secreted into surfaces such as the respiratory tract, GI tract, and reproductive tract. So this then would be aggregated nodules of the tonsils containing a secondary nodule, and here would be a primary nodule, right? There's no germinal center in the center, just like there is a germinal center here, so it's a secondary nodule. Here it's a primary nodule because there's no germinal center. Here's another primary nodule, no germinal center. Okay? So that's the tonsils. Similarly, we have aggregated nodules in Peyer's patches. Peyer's patches are these aggregated nodules that occur ostensibly in the ileum of the GI tract. And again, they can be formed and dissipate. They can be formed from loose lymphatic tissue that can then aggregate to form, in this case, they're primary nodules because we don't see a well-defined germinal center. So these are Peyer's patches, aggregated nodules in the ileum. And then in these Peyer's patches, sometimes we have what are called microfold cells. These are special and rare cells that can directly bind a pathogen such as a bacteria and pass it off to a macrophage that is readily immediately adjacent to a lymphocyte to immediately activate an immune response. So the microfold cells are acting as a short circuit, if you like, between the bacteria and the macrophage to stimulate B cell or T cell mediated immunity in the Peyer's patches. And then the third type of aggregated nodule is the appendix. Okay, here's the appendix. Has anybody had their appendix removed? Did they go through the belly button or the side? The side? You want to help me? Come up here and you can be my model. You're going to help me find where your appendix was, right? Stand over there. Okay. Put your right hand on your hip, right hip. Put your left hand on your belly button. Now, trace a line 1/3 of the distance from your hip to your belly button. Other way, from your hip to your belly button. From your hip to your belly button, this way. Start off with your hip. Yeah, right there. That should be where you had your surgery, right? Yes, see? Thank you. That is what we call McBurney's point. See that up there, McBurney's point? Right? So, when you go to have your physical diagnosis, if you have a stomach pain, right, and you go to the doctor and say, "Doc, I've got this pain. I don't know what it is." What he's going to do, he's going to start feeling around your abdominal area, and he's going to find McBurney's point right there, and he's going to kind of push gently on that. And if you squeal, he's going to think that, okay, maybe you've got an inflamed appendix, and then he's going to do additional stuff to see whether you have appendicitis and whether you need to have surgery. Actually, when I was a kid, people were having their appendix removed all the time. In this day and age, if they get it early, you don't have to have surgery necessarily. They can just give you massive quantities of antibiotics and that will get rid of it some of the time, but not always. But if it ruptures, you've got to have surgery quickly. Because actually, a distant relative of mine, she had ruptured appendix and she died from it. And they took her to the hospital and she died. Question in the back? It what? Probably all types of bacteria, good and bad, because it's kind of like a little diverticulum. So they really don't know why it's there, but it's kind of a trap for bacteria, good and bad. So if the bad take hold, then it can cause rupturing. But yeah, good, because your entire GI tract is lined with good bacteria. Those commensals, remember the very first slide of the previous lecture, we talked about commensals, which are good bacteria that help your body digest certain foods and things. So yeah. Okay. So here is a picture of the appendix in a younger person and an older person. And the thing to note is the amount of lymphatic tissue in the appendix. So in the younger person, there's quite a bit of lymphatic tissue. In the older person, you can see most of it's gone, right? So this is consistent with that theme where we see as an individual gets older, the amount of immunological tissue that's present in their body progressively declines, okay? So you remember in the bone marrow, you get red marrow and yellow marrow, so the red marrow is replaced with yellow marrow, which is fat cells. In an older person, in the appendix, you get replacement of some of the lymphatic tissue with adipose tissue. We'll see the same thing in the thymus when we do that momentarily. So that was aggregated nodules. So recall, nodules are not encapsulated with connective tissue. They may be partially encapsulated, such as the tonsils, but not fully encapsulated. So then we have specific organs that are encapsulated. So I showed you this slide in the bone marrow slide. So basically it's encapsulated because the bone marrow is lined by endothelial cells, okay, inner endosteum, right, things like that, which can basically line the inner cavity. So I'm not going to go over this because we know all of the different components of hematopoiesis that give rise to the bone marrow cavity. Then the thymus, remember the thymus is the location where T cells mature. So here is our bilobed thymus and its location in the chest area. And if we look at the thymus, it is fully encapsulated. So on the periphery, there is a connective tissue capsule which can branch to form trabeculae, dividing the thymic tissue into what we call lobules. This is a lobule from here to here. And the lobule contains at its outer perimeter a cortex of immature T cells and a medulla containing some macrophages, Hassel's corpuscles, and maturing T cells. So here's a schematic of a lobule, connective tissue capsule, a septa of connective tissue, and in the cortex are the T cells maturing. Some of them are released, and Hassel's corpuscles are the epithelial reticular cells, which form a framework upon which the T cells reside. So it turns out that the process of thymocyte maturation is very inefficient. So in the thymus, the T cells are undergoing that somatic recombination event. Remember, on the surface of T cells we have a T cell receptor that has that variable portion. So if that T cell is recognizing a self-antigen, right, which would then cause some sort of autoimmune response, that T cell is phagocytosed by a macrophage and destroyed. Whereas if that T cell that is undergoing somatic recombination does not recognize a self-antigen, then that T cell will survive and be released into the circulatory system, into the blood, if you like, so that it can act out immune surveillance. Now, this is very inefficient because 95% of the T cells in the thymus that are undergoing maturation are self-recognizing, and so 95% of these T cells are destroyed by macrophages, whereas only 5% are non-self clones, and so only 5% are released into the circulatory system. So it's very inefficient, okay, very inefficient. 95% are destroyed, 5% make it. This is actually a very good argument, in my belief, against the concept of intelligent design. Have you heard this intelligent design concept? People that don't want to believe in evolution have come up with this intelligent design where they said things didn't arise by evolution, there was an intelligent designer that made things as they are, and that's it, okay? Well, if you're an intelligent designer, would you design something that's 95% inefficient and 5% efficient? You'd do it the other way around, right? You'd have 95% or 100% efficient and 5% inefficient. So to me, this is a biological example of an argument against the concept of intelligent design. Just throw that out there for your consideration. Okay, so in the thymus, we also have a blood-thymus barrier. We don't want blood-borne components of the immune system having ready access to those thymocytes as they're undergoing somatic recombination, because if they're recognizing them early on during somatic recombination, they might destroy them. So we have a blood-thymus barrier that prevents formed elements of the blood from readily coming into contact with the thymocytes in the cortex of the lobules, preventing an interaction. So there's a couple of locations in the body where we have blood barriers, like there's a blood-thymus barrier, there's a blood-brain barrier, there's a blood-testis barrier. That's the three that come to mind. And in all cases, you don't want formed elements of the blood coming into contact with cells in those regions, causing an immune response. So you'll recall in the thymus, we have epithelial reticular cells that form a microframework upon which the lymphocytes can reside and undergo somatic recombination to select out self and non-self clones. So these epithelial reticular cells, as they die, can be curled up into a little ball or a bundle here, which then we call Hassel's corpuscles. So these Hassel's corpuscles are a unique histologically distinguishing feature of the thymus that allows us to identify the thymus as a component of the immune system and not something else. So we can say, okay, because we see Hassel's corpuscles, this is thymus and not a lymph node or spleen and so on. Okay, so here's a schematic of the histopathology of the thymus. Here's our cortex where the T cells are undergoing selection, somatic recombination, then they're released. The mature ones are released into the vasculature, the rest are phagocytosed by macrophages. Here's our blood-thymus barrier, and more typically the Hassel's corpuscles we'll see down in the medulla of the thymus. Now, as the thymus gets older, it also undergoes involution. So you can see here in a younger person, the amount of T lymphocytes in the cortex and the medulla is relatively large and the amount of fat is limited. But as a person undergoes aging, they undergo immunosenescence where as you approach old age, here I am right here at 60, you can see I haven't got a lot of T cells and I've got a lot of fat in my thymus, looking like more, even worse than that, right? So this just, again, highlights the fact that as a person gets older, the components of the immune system undergo immunosenescence. So we've seen that in bone marrow, where the red marrow is replaced with yellow marrow, right? We've seen that in the appendix, right, where the younger person had quite an abundance of lymphocytes, and then the person, I think was 36 years old, had very few T cells. Now we see it in the thymus, where again, much of the T cell component of the thymus is now replaced by adipose tissue. So this is a general theme that occurs throughout life. And that's why people, as they approach, like really old age, like in their 70s and 80s, get infections much more easily than a younger person, right? So that's why actually if a person, I think over the age of 65, when they get immunized for the flu vaccine, they give them a much stronger shot. I think it's 10 times the amount of immunogen than I received when I had my flu shot last month. And that's because they really want to boost the immune system as much as they can to fight against the virus. So if you have, an aged relative, grandma, granddad, and you've got the flu or a cold, you shouldn't go and interact with them because you can infect them and they could be terminal, right? Because their immune system is very much suppressed down here as opposed to yours, which is like in this range here for most of you, I would think. So just be, and that's why in the United States, I believe, I think it's 30,000 people die of the flu each year, mostly the elderly or very young. That's because those two populations have very much compromised immune systems. The elderly down here and the young, their immune system hasn't fully matured yet, okay? So, you know, you want to stay away from older people if you are ailing from what you think is some sort of infection, flu, particularly flu, but also a cold, because they are so much more susceptible to catching it and not being able to suppress the infection. Okay, specific organs. Okay, carrying on now to the lymph nodes. So the lymph nodes are a filter of the lymphatic vessels. And the lymphatic vessels basically drain the connective tissue, and it's unidirectional. They drain, they're blind ending, and they drain the connective tissue back eventually to a large venous vessel like the vena cava, okay? And they're concentrated in axillary lesions, such as around the armpits and the groin area and along the spinal column. There's bundles of these lymph nodes. Now, because the lymph nodes and the lymphatic vessels are relatively porous, they're actually already a route for tumor cells to circulate through the body. So, for instance, if you have breast cancer and you have the primary tumor removed, quite often what will happen is that the surgeon will biopsy the lymph nodes in and around that primary tumor, because what he's wanting to see is whether any of the tumor cells have migrated from the primary tumor to an adjacent lymph node, okay? Because if that's happened, then that's not such a great prognosis. What he's hoping to see is no tumor cells in the adjacent lymph nodes, because then that means, hopefully, that they got the whole tumor during the surgery to remove the primary tumor. So, and surgeons can give a prognosis on the outcome of following surgery of a patient's ability to recover based upon the degree to which lymph nodes and the distance from the primary tumor of those lymph nodes are positive for tumors. So if there's no tumor cells in the lymph nodes around the primary tumor, that's a good prognosis. If there's like 1 out of 10, that's not as good as none, but it's better than if all 10 lymph nodes were filled with tumor cells, because if all 10 were filled with tumor cells, that means that the tumor cells have spread to a great deal from the primary tumor. So they've obviously studied this for many years, and they've got charts, and you know, if it's 10%, 50%, 100%, they can give you a much clearer prognosis as to, you know, what you can expect going down the road following your tumor. And that may also inform the surgeon as to whether they need to do follow-up radiation or chemo or endocrine therapy, something like that, based upon what they see in the lymph nodes surrounding the primary tumor. So the tumor then is very useful and is biopsied to look at tumor cell metastasis from the primary. So here's our lymph node right here. Okay. Now the lymph node is the filter of the lymphatic fluid. Okay. So if we look at a schematic of the lymph nodes, kind of like bean-shaped if it's full, the lymphatic fluid comes in on the surface of this bean through the afferent lymphatic vessel. Then the lymphatic fluid will drain into the subcapsular sinus. Then it will pass down the trabecular sinus into the medullary sinuses, and then it will exit the efferent lymphatic. So then the lymphatic fluid is draining in one direction from the afferent through these various sinuses down to the efferent lymphatic, which is in the hilum, or the indentation. Conversely, blood comes in through the hilum, makes its way in small blood vessels into the paracortical region, does a U-turn, and then flows back out through the hilum. within the lymph node, sometimes we can see nodules. Okay, so the node itself is encapsulated, right? Because we've got this capsular sinus, which is connective tissue capsule. So the lymph node is encapsulated, and inside the node we'll see nodules. Remember, nodules are non-encapsulated aggregations of lymphatic tissue. And we see these nodules in the more towards the cortex. Okay, and that's where we'll have an aggregation more of B cells. Here and also in the medulla. And in between the medulla and the lymph nodes, in nodules, in this paracortical region, we tend to get an aggregation of T cells. So B cells and T cells are differentially aggregated in different parts of the lymph node. Okay, B cells in the cortical nodules, T cells predominantly in the paracortex of the lymph node. Now, lymphocytes make their way into the lymph node from the blood vessels by passing across what we call post-capillary high endothelial venules. So most blood vessels, or all blood vessels except these guys, are lined by a squamous layer of endothelial cells. Okay? Squamous meaning flat, long, elongated. However, in the lymph node, after the capillaries, we have special capillaries that have the endothelial cells that are more cuboidal, right? So this is why we say they're high endothelial, because they're no longer squamous, they're high endothelium. And as the blood passes into these high endothelial venules, the lymphocytes squeeze between these endothelial cells and then exit out of these blood vessels into the parenchyma of the lymph node. So that's how the lymphocytes get out into the parenchyma of the lymph node itself, where then they may come into contact with pathogens such as bacteria draining through those sinuses. Remember, there's the subcapsular, trabecular, medullary sinuses. So then the lymphocytes have got out of the blood vessels into the parenchyma of that loose scaffolding of the lymph node, looking out for any pathogens that are draining through the lymph node. Because remember, the lymph node is the filter of the lymphatic fluid. Okay, and then the last organ that is encapsulated is the spleen. Here is the spleen over here. So the lymph node that we just looked at is the filter of the lymphatic fluid coming from the lymph vessels. So the lymph node, the filter of the lymphatic fluid. The spleen is the filter of the blood, okay? Spleen is the filter of the blood. It also plays a role in an immune response because you've got, in addition to red blood cells, you've got lymphocytes in there that are undergoing immune surveillance. Because it's so large, it's also thought as a storage organ for platelets. Okay, it's storing platelets for a sort of a blood clotting response if necessary. And also the spleen is capable of extramedullary hematopoiesis. So when we say extramedullary, medullary is referring to the bone marrow. So extramedullary, outside of the bone marrow, hematopoiesis. So again, the spleen, just like the liver, is capable or has this genetic memory of its second trimester fetal life. Remember second trimester, the hepatospleenothymic phase, hepato, liver, spleen, spleno, now is what we're talking about, hepatospleenothymic phase of hematopoiesis. So the spleen has this genetic memory of its second trimester fetal life where if called upon, it can reactivate hematopoiesis. Again, if the bone marrow is damaged in some way, the spleen can take over and undergo some residual hematopoiesis in the spleen. And it happens in certain diseases. So if we look at the structure of the spleen, it's encapsulated connective tissue on the outside, and we describe it, the spleen as containing red pulp and white pulp. So red pulp contains erythrocytes, red blood cells, no nuclei, they're stained pink, right, no nuclei. The white pulp contains an abundance of leukocytes, nucleated components of the blood. So the nuclei stain purple with hematoxylin, so that's why you get this purplish color of the white pulp. White pulp is referring to white blood cells. Red pulp, white pulp. So if we look at the vasculature and the circulation of blood entering the spleen, it comes in through a large systemic artery, and then it passes through what is called the central artery of the white pulp. And then as it passes down that central artery, it goes out smaller blood vessels to the margins of that white pulp, and then it makes a U-turn and flows back into the central artery, and then it exits the white pulp into the red pulp. The red pulp contains large sinusoids, which are kind of like large porous veins, where the red blood cells are examined for age by macrophages, and if they are aged, they can be removed by macrophages. Otherwise, they can pass through and then pass back into the hepatic portal system. So schematically then we can see blood entering the central artery or arteriole. It's kind of on the borderline between an artery and an arteriole, into the white pulp containing lymphocytes. That blood, some of it goes to these marginal zone sinuses shown here, where there will be macrophages that may interact with any pathogens that's flowing through these sinuses, and if these pathogens are found to be foreign, it may cause an immune response, and you may get a secondary nodule formed within the peripheral white pulp, shown here. Then that blood can flow back to the central artery. So out here in the periphery, most of the lymphocytes are B lymphocytes, peripheral white pulp B lymphocytes. Right around the central artery, there's also a sheath or a collar of lymphocytes, predominantly of the T cell variety. So here we have what is called the periarteriole next to the artery lymphatic sheath, like a collar or a sleeve around this artery containing T cells. So then in the white pulp, we have T cells immediately adjacent to the central artery and B cells more peripherally located outside of the periarteriole lymphatic sheath. Okay? So then the blood may pass back into the central artery, then back out into the sinuses and the sinusoids, where it will be examined for aging of the red blood cells. So when these red blood cells pass into these sinuses, shown here, sometimes they can pass into and out of these sinuses through slits between the adjacent endothelial cells. And these sinuses sometimes, or for the most part, are discontinuous with the vasculature. So these red blood cells can pass between the endothelial cells out into the parenchyma, then pass back in, and then back into the sinus. Okay, this is called an open circulation. If there's a continuity between the blood vessel and the sinus, that's closed circulation. So what happens is the red blood cells can pass out into the parenchyma, and then they want to squeeze back into the sinusoid through these slits shown here. So here's our sinusoid. Here are the slits between the endothelial cells. And here you can see how these red blood cells are squeezing right here between the slits of the endothelial cells in the sinusoid. If these red blood cells are less than 120 days of age, they're still relatively flexible. Remember, we talked about the flexibility of red blood cells when we talked about red blood cells as part of hematology. So if they're still flexible, less than 120 days or thereabouts, they can squeeze back into the lumen of the sinusoid and then exit into the veins. If they're relatively old, okay, let's say 120 days, or if they're a sickle cell, they've got this change in shape that renders them relatively inflexible, then adjacent macrophages are going to attach these red blood cells and phagocytose them and destroy them. So then the spleen is considered the filter of the blood because it's filtering aged red blood cells, or in the case of sickle cell anemia, sickle cell shaped red blood cells. So the spleen is the filter of the blood. Okay, there you go, a little bit quicker. So which of the following accounts for most of the lymphoid tissue in the body. Most of the lymphoid tissue. Did I hear somebody say something? Most of the lymphoid tissue is actually just the loose mucosa-associated lymphatic tissue, because the GI tract, the respiratory tract, the reproductive tract are quite extensive, and they're completely lined by loose lymphocytes throughout. So if you counted up all of those lymphocytes in all of those locations, it would actually amount to quite a lot of lymphatic tissue. And in fact, in one of those early slides, I think I said it accounts for 85% of the lymphatic tissue of the body. Let's take a 5-minute break, and then we'll do the lab, if that's okay. They can, yeah.
They can.
Okay, shall we take our seats? Okay, lab time. Let's look at some examples of all of these different cells and tissues that we have been talking about. So first, you need to be able to identify the different cell types, tissues, and organs discussed in the 4 lectures. Okay, and then the thing that you want to do is look for distinguishing features that allows you to say it's this particular tissue or organ and not something else, and then know the unique function of these different structures. So if we look at the thymus from a younger individual, you can see this piece of tissue has quite a bit of hematopoietic tissue associated with it. So, of course, thymus we consider to be a central lymphoid organ because it is the location where we have the final maturation of T cells and release into the circulatory system, as opposed to B cells which mature in bone marrow. Both of these then will exit into the peripheral lymphatic tissue, where they may reside as loose connective tissue, aggregated in mucosal surfaces, or spleen and lymph nodes filtering lymph and lymphatic fluid. Okay, so loose lymphatic tissue, lymphocytes. So many of these nuclei that we see here underlying this section of the intestine are simply loose lymphatic cells of the lamina propria in this location. Any questions? And of course, if those lymphocytes, B lymphocytes, become activated, we recognize plasma cells which are producing lots of antibody. So we know how to identify a plasma cell because typically it has an eccentric nucleus. The nucleus may have a cartwheel arrangement of the heterochromatin, and of course it has an abundance of rough endoplasmic reticulum producing lots of immunoglobulins. Any questions? Okay, so here we have in comparison a plasma cell next to an eosinophil. So the plasma cell again has an abundance of rough endoplasmic reticulum. The eosinophil we should be able to recognize because it has these specific granules that contain this stripe. Okay, so the internum as opposed to the externum of the stripe. Any questions? Okay, so sometimes the loose lymphatic tissue can aggregate. So here's the loose here. Lymphocytes can aggregate as a nodule. It's a nodule or follicle. Does not have an encapsulating connective tissue around it. And of course this would be a secondary nodule because we can recognize a mantle, okay, and the lightest staining central area. So this would have more plasma cells producing antibodies as opposed to the mantle, which would be the still unactivated lymphocytes. Questions? So what sort of lymphocytes, would they be B cells or T cells in this area? B cells, okay? B cells in the mucosa and tissues, T cells in the blood, okay? Differentially expressed. So here's a high power view of this secondary nodule or follicle. You can see again the mantle, which would be the unactivated B cells, and the germinal center, which is contain more plasma cells and maybe a few other components of blood. Any questions? Okay, so right in the germinal center again, you can see now some cells have more cytoplasm because they've become activated to produce antibodies. Tonsils, there's a lot of them surrounding the oral cavity and the respiratory tract. First surveillance, if you like, of pathogens as they enter into your body. You do not need to know any of these tonsils, the names of any of them. If we look at a cross-section, histological section through tonsils, we can see there is a connective tissue capsule underneath, and sometimes this connective capsule separates different pieces of the tonsils, but on the surface we have stratified squamous epithelium, non-keratinized epithelium. So that's not connective tissue, that is integument, i.e. skin. So it's not considered connective tissue capsule. So then we say tonsils are not an encapsulated organ, okay? Not encapsulated. And of course, within the parenchyma of the tonsils themselves, we can see some secondary nodules that are formed, right? So here you can see the germinal center on a thin mantle. In many cases, these secondary nodules are formed within this aggregated nodules of the tonsils. Any questions? Okay, Peyer's patches of the ileum, again the same concept. We can see now these, looks like halfway between a primary and a secondary nodule, has formed within the submucosa of the ileum here. And these may thin out and increase depending upon the state of infection. Any questions? Yes. The outer layer of a secondary nodule, because that is the B cells that have yet to be activated, whereas the germinal center, which stains lighter, has more plasma cells in it. The B cells differentiated into plasma cells. The plasma cells have more cytoplasm containing rough endoplasmic reticulum. So because you've got more cytoplasm relative to the nucleus, it's going to be a lighter staining area, right? B cells, mostly nucleus or thin rim of cytoplasm. Plasma cells, a lot of more cytoplasm, so it's going to stain lighter in the germinal center where we have many more plasma cells. Other questions? Yes. No, it's underneath. So this is the lumen of the ileum. Here's the lining epithelium. Here's the submucosa out here. So it's all underneath. It's inside the body. Other questions? Okay, so here's our M cell in the ileum. Remember, these M cells can attach bacteria and viruses and hand them off directly to a macrophage, which will be in the vicinity of the lymphocytes to short circuit activation of the lymphocytes. Okay, so it's just a way to speed up an immune response in the GI tract. Here's our appendix again. You can see loose lymphatic tissue here. And then here we have an area of aggregated nodule containing a germinal center and a mantle. Loose secondary nodule, loose secondary nodule, and so on. It's transitory. It can come and go, develop and dissipate. Any questions? And again, comparing younger and older individuals of the appendix, shown here, a young individual has more lymphatic tissue than someone that is considerably older in age. Bone marrow, we know all about bone marrow. We spent a couple of lectures on bone marrow. Primary location for production of formed elements of the blood, okay? Thymus, bilobed, shows the location in the chest area. And you can see how abundant this tissue is in this younger individual. It's encapsulated all the way around the capsule, which can branch to form these trabeculae, separating out these different lobules. And in the cortex of the lobules, we'll have the immature T cells undergoing the somatic recombination. Then these thymocytes, as they mature, become T cells. Some of them will be selected out, some of them will make the rite of passage and enter into the circulatory system. And in the medulla, also sometimes at higher power, we can make out these circular structures of the epithelial reticular cells. Any questions on this one? So here's a higher power view: capsule, lobule, cortex, medulla, and now you can make out some of these Hassel's corpuscles. Just make them out here at a low power view. Questions? Okay, and then at much higher power view you can make out the Hassel's corpuscles. So the Hassel's corpuscles are the distinguishing feature that allows you to say that this is thymus as opposed to lymph node or spleen or aggregated nodule or something else, okay? That's the unique feature of the thymus, the Hassel's corpuscles. You won't see them elsewhere. Any questions? Okay, younger and older thymus, you can see here abundant T cells in the older individual, replacement with adipose tissue. Okay, so again, decline in the capacity of the body to mount an immune response because of a gradual decline in lymphatic tissue. If it's in the bone marrow, replacement of red with yellow bone marrow. If it's in the appendix, right, where you get replacement of the lymphocytes with adipose tissue. Thymus, same sort of thing. Okay, gradual decline with age of the amount of lymphatic tissue in organs and tissues of the body. Okay, questions? Okay, so here's our lymph node shown here. And at low power you can see this bean-shaped structure. Subcapsular sinus is going to drain into trabecular, then medullary sinuses, and then exit out the hilum down here. Blood vessels come in the opposite way, come up to the paracortex, do a loop, and then exit as well. So in the cortex we have these nodules, which appear to be secondary nodules. In the paracortex, which is mostly beneath these cortical nodules, we will have the high endothelial venules and the enrichment of T cells in the paracortex. So more B cells in the cortex, more T cells in the paracortex. Questions? Okay, here it is again. The cortex, follicles or nodules in the cortex, mostly B cells. In the paracortex, mostly T cells. Any questions? And then, of course, we have the sinuses, subcapsular and trabecular sinuses, shown here at a high power view. And in there we also have reticular cells, which are staining up with a silver-based stain. These reticular cells form a framework upon which all of these B cells can aggregate, on the lookout for any pathogens that may be draining through the lymph node. Okay, here's shown again with the reticular silver stain. Here's the subcapsular sinus, trabecular sinus, medullary sinus. Fluid is draining through here. And Up here you've got B cells in the cortical regions, T cells in the paracortical region, and in here you'd also have some high endothelial venules. Any questions? Okay, so the reticular cells are made up of, contain an abundance of reticular fibers. Remember, reticular fibers are a specific type of collagen. They are type 3 collagen, correct? And they stain up well with a silver-based stain. So they'd stain black with a silver-based stain. So reticular fibers in reticular cells forming a scaffolding throughout the lymph node, upon which lymphocytes can hang out looking for any pathogens that may be draining through this scaffold. Any questions? Okay, so here is how high endothelial venules in the paracortical region, right? Cortex, medulla's down here. Paracortex enriched in T cells. Here are these high endothelial venules magnified even higher. You can see they're more or less cuboidal, not so much squamous. So then the lymphocytes are going to come in through this vessel, squeeze through these high endothelial venules, and exit into the parenchyma of the lymph node itself. Any questions? So the high endothelial venules are a unique feature of lymph nodes. So if you were shown something that appeared, lymphatic, and you saw high endothelial venules, you would know it had to be a lymph node, because you don't get high endothelial venules in spleen or thymus or aggregated nodules and so on, okay? So that, again, is a unique histological feature of lymph nodes. Only in lymph nodes do you get high endothelial venules. Spleen is our last organ. Okay, so the lymph node was the filter of lymphatic fluid. Spleen is the filter of the blood. It's a large encapsulated organ. And here's the capsule, connective tissue capsule. Red pulp contains predominantly red blood cells. White pulp predominantly contains leukocytes of the lymphocytic variety filtering through here. Any questions? And at a high power view, we can see the white pulp here. Now here, note the what is called the central artery or arteriole. Note the one thing about the central artery is that it's never central. It's always eccentric off to one side. Okay? So here's our central arteriole. And around the central arteriole would be the periarterial lymphatic sheath of T cells. And then out here would be the marginal zone sinuses. So outside of the pals would be predominantly B cells in the rest of this white pulp area of the spleen. Any questions? Yes? It's not easy to see the marginal zone sinus here, but it's going to circumscribe this darker stained area of lymphocytes. So it's going to be like here, like maybe here. These are marginal zone sinuses. Okay, circumscribing the entire structure. I had a schematic of it in the previous hour's lecture. You can't really make them out too clearly. Other questions? Okay, so here's our central arteriole. Again, it's not central, okay? Here's our marginal zone sinus. We'll circumscribe this piece of white pulp out here. So right around the pals we will have T cells, and elsewhere in the rest of this white pulp we will have predominantly B cells. And since we have a germinal center, that would suggest some sort of activation of B cells to form plasma cells producing antibody, because they're plasma cells, they have more abundant cytoplasm. That's why this area in the center here is staining a lighter color, more cytoplasm of the plasma cells as opposed to not so much cytoplasm of the B cells. Questions? Okay, so then beyond the white pulp, the blood drains into the red pulp containing predominantly red blood cells. So here you can see these sinuses, and here you can see red blood cells, and these nucleated cells, macrophages, there's too many of them to be leukocytes in blood, okay? There's way too many. So these are macrophages that are hanging out, waiting for any red blood cells to come along that show signs of aging, okay? So then here's our sinusoid. Here are the slits between the adjacent endothelial cells. Here we have some macrophages looking for aged red blood cells so that they can capture them and phagocytose them. Any questions? Now, of course, the spleen, being the filter of the blood, is also a location for recycling of iron. So iron is removed from heme, of the hemoglobin in the red blood cell. So if we stain for iron with this Prussian blue stain, you would expect to see an abundance of iron in the spleen, because then that iron is recycled and reutilized throughout the body. Questions? Okay, there you go. That's it for the lab. Here's our question. In the spleen, so we know we're looking at the spleen, the area around the structure shown, so you see the arrow, the area around this arrow is enriched in what cell type? What do you think it is? Enriched around this structure. Okay? It's going to be T cells. T cells are enriched in the periarterial lymphatic sheath. Okay, there we go, guys. That is for today. And then next Wednesday, we are doing the cardiovascular system. What was A? Well, the red blood cells, because this is the white pulp. So the red blood cells are in the red pulp.